So a couple of weeks back, I introduced a concept of a diet called the Emergence Diet. And it's a pretty high protein diet. The concept behind it is pretty simple. It's just, what do grizzly bears do in the spring when they are coming out of torpor? And the period of time when animals come out of torpor is called emergence. And so this is a different study than I've shown about what grizzly bears actually eat. And this dark area, this is in male grizzly bears, this is in females, this is ungulates. And so in the spring, the period from May through July, which is really the emergence time of year, the bears are eating a lot of elk and they're eating a lot of grass. And you can see that grizzly bears killed an elk calf every 4.3 days during June. Elk accounted for 84% of all ungulates consumed. And so the grizzly bears are eating essentially lean meat. Elk is very lean. So they're eating lean meat and they're eating plants, green plants. So I haven't talked a lot about protein. It's a controversial subject and it's complicated. And so I don't like to talk about things if I haven't had a lot of time to think about them. Even within the kind of keto community, the amount of protein that you should eat is kind of controversial. So this screen here is the PE diet, and this is Ted Naiman's diet. And he talks about the amount of protein versus the amount of energy. So energy means starch and fat, right? Whereas protein, he says, is more satiating. And you can see by the diagram here, will make you lean. Yet there are other voices in the community, such as Amber O'Hearn. And you can see there her blog, Eat Meat, Not Too Little, Mostly Fat. And I know people who say that if they go onto a Ted Naiman style high protein, low carbohydrate diet, they will gain weight on that diet. But in human studies, it seems that in general, protein is helpful for weight loss. So here it says several meta-analyses of shorter-term, tightly controlled feeding studies showed greater weight loss, fat mass loss, and preservation of lean mass after high-protein energy restriction diets than after low-protein energy restriction diets. However, there is a danger here. Branched-chain amino acids, valine, leucine, and isoleucine, are a type of amino acid that is high in animal protein and high especially in dairy protein. And interestingly, branched-chain amino acids, if you look at the plasma circulating levels, in obese humans are higher than in lean humans, and high circulating levels of branched-chain amino acids look like they're associated with insulin resistance, and in fact, there's evidence that they contribute to the development of obesity-associated insulin resistance. And that paper shows this. You can see that in the so there's valine, leucine, and isoleucine on the right there. Those are the amino acids, and you can see the levels are higher in obese humans than they are in lean humans of all three of those branched-chain amino acids, as they call them. And you'll also notice that something called alpha-ketoglutarate is lower in obese humans, and we are going to come back to that concept. So this is, again, another paper. Recent studies implicate a strong association between elevated plasma branched-chain amino acids and insulin resistance. And so if we think about the emergence diet, it's like, okay, well, so I have elevated branched-chain amino acids. It's contributing to insulin resistance. But should I be eating all of this lean meat, which is obviously going to provide a lot of branched-chain amino acids, right? And it goes on to say these data demonstrate a pivotal causal role of a branched-chain amino acid catabolic defect and elevated abundance of branched-chain amino acids and branched-chain keto acids in obesity-associated insulin resistance. What they're saying here is that for some reason, obese humans can't break down. Catabolism means breaking down. So obese humans, the problem is not necessarily that they're consuming too many branched-chain amino acids. It's that they can't break down the branched-chain amino acids. So if you've followed any of my work, that I like to use the analogy that obese humans are essentially in a similar metabolic state as animals in torpor, which is the state that mammals go into in the winter when they lower their metabolic rate to preserve their fat mass for winter. An amazing thing about torpor is that animals go all winter long. They're not eating, right? So they don't consume any protein. And yet they emerge in the spring with almost the same amount of muscle tissue as they had in the fall. So their muscle tissues don't atrophy over the winter. This, of course, makes a lot of sense. Branched chain amino acids are actually very anabolic. If you have a lot of circulating branched chain amino acids, they can cause you to build muscle tissue. So in torpid animals, what this study is saying, winter depleted proteins included those that degrade branched chain amino acids. So in torpid animals, they lower the amount of the enzymes that break down the branched chain amino acids. And this is presumably one of the things that they're doing to help them preserve their muscle mass. And interestingly, we see the same thing in obese humans. And of course, torpid animals in the winter are also insulin resistant because they're obviously not eating starch. They don't have any incoming glucose. So you could see why the branched chain amino acids would have evolved in a way that if you're not breaking those down, you're preserving muscle mass and you are insulin resistant. This is another paper in the torpid animals. It says, the pattern we observed likely reflects reduced protein and amino acid catabolism during torpor and its resumption during periodic arousals to euthermia, right? And so when the animals are getting out of torpor, they're cranking back up their ability to break down the branched chain amino acids. And so the flip side of this is that in the lab, so this paper is called Restoration of Metabolic Health by Decreased Consumption of Branched Chain Amino Acids. So in the lab, if you take mice and you put them on a Western diet, they will become obese. If you look at this graph here, in the beginning, there are 4 groups of mice. They've all been on a Western diet for a while to make them obese, and then they're switched. This black line continues on the Western diet. The purple line is the Western diet supplemented with branched chain amino acids. And these orange and blue lines here, go Knicks, these are diets that are either limited in total protein or specifically the blue line is restricted branched chain amino acids. Right, so you can see mice that were fat on a Western diet, but then switch to a diet that's still a Western diet, but with limited branched chain amino acids, rapidly lose weight. Furthermore, they rapidly lose fat mass specifically, although they do lose a little lean mass, but mostly they lose fat mass. So these are on a Western diet with restricted branched chain amino acids only. This is after 2 weeks. This is after 10 weeks. And if you compare it to the white line, which is the control mice, they're actually eating more. And this is their spontaneous activity. So they're moving less. And so those mice lost all of that weight from the Western diet by eating more and moving less than the control mice when they were put onto a branched chain amino acid restricted diet. So this is back in humans. And what you can see is this is thermogenic effect after a meal. So humans were given different meals. In this meal, they were fed carbohydrates, and you can see it's 2 megajoules. That's like an amount of calories. I'm not sure off the top of my head exactly how many calories that is, but this line here is baseline. This is the baseline metabolism of the person. So when carbs are consumed, you see this increase in metabolic rate. So carbs are thermogenic when you're burning them, and these are hours down here. So this is about a 3-hour peak in metabolic rate after you consume carbs. This is protein here. This is the same amount of calories. You see it's 2 megajoules here, it's 2 megajoules here. So this is protein. You see this massively increased amount of thermogenesis in a high protein meal versus a high carbohydrate meal. You can see that in obese humans, so this is only half the amount of calories, but it's consistent in all these graphs. The obese humans have much less of a thermogenic response in response to a high protein meal. And this is the case at both levels of caloric consumption. It's also the case for carbohydrate consumption. The obese humans just don't quite have the same thermogenic response to those proteins. And so in that video, the Emergence Diet, I talked about how one of the things that the animals do when they're trying to get out of torpor, when they're trying to get their metabolic rate going again, is they surge 3 things. They surge pyruvate, they surge succinate, and they surge alpha-ketoglutarate. In a lot of past videos, I've talked extensively and on my blog about pyruvate and about succinate. For the rest of this time, we're going to focus on alpha-ketoglutarate and what its role is in helping to break down those branched chain amino acids. And so this paper is talking about one of my favorite topics, which is ROS production. And so pyruvate, and I've talked about this extensively via pyruvate dehydrogenase, produces ROS as it's being broken down. And so that one there, BCKDH, is branched chain keto acid dehydrogenase. And as you might imagine, that one is used in breaking down those branched chain amino acids, and it also produces ROS. That one there breaks down alpha-ketoglutarate, and guess what? It also produces ROS. And interestingly, all three of these enzymes use lipoic acid as a cofactor. Lipoic acid is a very redox active molecule. It has two sulfurs. And in the process of moving the electrons around, these enzymes all release significant ROS, and ROS is the throttle to your metabolism. I've talked about this in a lot of past videos, including the ROS theory of obesity. This is a simple drawing of the catabolism of branched-chain amino acids. So we're looking at breaking down those branched-chain amino acids. In the breakdown of all of these branched-chain amino acids, this one here is this is a transaminase. And what it does is it converts alpha-ketoglutarate to glutamate. And that allows these branched-chain amino acids to be broken down. If you can remember from that chart that I showed, in obesity, these branched-chain amino acids are high, alpha-ketoglutarate is low. And so alpha-ketoglutarate is a limiting factor in this first step of breaking down those branched-chain amino acids. I drew in kind of the rest of, this is the TCA cycle, of course. This is where we burn everything, carbs, protein, fat. It all goes ultimately through the TCA cycle. And I've drawn in, here's succinate dehydrogenase. Here's where the alpha-ketoglutarate dehydrogenase comes in. And pyruvate goes through pyruvate dehydrogenase. This number 2 here is the branched chain keto acid dehydrogenase. And so this breaks down these branched chain keto acids, as they call them, once they are deaminated. And so all of these things circled in yellow produce reactive oxygen species. And so this is presumably one of the reasons why protein is highly thermogenic, because in this first step, the branched chain keto acid dehydrogenase is producing ROS, but then you can see a lot of these get converted to succinyl-CoA, and that, of course, succinyl-CoA becomes succinate, and that goes straight into succinate dehydrogenase. So they're producing ROS here, and they're again producing ROS right here. The alpha-ketoglutarate is helping with this process. It's necessary, of course, for this first step, the transaminase. But alpha-ketoglutarate, if it's not used for transamination, of course, it goes straight into this alpha-ketoglutarate dehydrogenase, and it produces ROS there, right? And so now think about those hibernating animals that are surging out of torpor. Well, they're releasing pyruvate, which is creating ROS here, and they're releasing succinate, which is creating ROS here, and they're releasing alpha-ketoglutarate, which is turning on this branched chain amino acid metabolism, which is producing ROS here, and then it's converting these branched chain amino acids into succinyl-CoA that is also getting converted to succinate and producing ROS here. So those three things together, pyruvate, succinate, and alpha-ketoglutarate, just really turn on that thermogenic machine. But there's a couple other factors that will affect your ability to break down those branched chain amino acids. And so this is the branched chain aminotransferase. This is the thing that converts alpha-ketoglutarate to glutamate. It can be turned off by acetylation. And if you listen to any of my previous stuff, you know that acetylation is caused by high NADH to NAD plus ratio. And producing ROS is one of the ways to get rid of some of that NADH and to provide more NAD plus. And so this is the first step in that thing. Interestingly, and I've talked in past videos about this, specifically meat and potatoes, this step uses active vitamin B6, which is also known as PLP. Vitamin B6 is another thing that is typically low in obesity, and this step is blocked by acetylation, and obviously it uses alpha-ketoglutarate. So in obesity, you typically see acetylated enzymes, and that's due to a reductive stress or a high ratio of NADH to NAD+. You see low vitamin B6 and you see low alpha-ketoglutarate. And so you can see why these branched chain amino acids are not being burned in obesity. This is the next step when we get to that branched chain keto acid dehydrogenase. Again, it is blocked by NADH, which is his end product. So that's classic end product inhibition of the enzyme, right? So now we're down here at step 2. This is the step that actually produces the ROS. And like I said, this enzyme requires lipoic acid, and lipoic acid, when it's produced, needs methyl groups. It actually needs SAMe to produce that lipoic acid. Obese humans have low levels of SAMe, and they have high levels of NADH. And so NADH blocks this step. So again, in obesity, you have low lipoic acid, you have high NADH, and all of this is contributing to this buildup of branched-chain amino acids and insulin resistance, etc. Again, just to summarize, why are branched-chain amino acid levels high in obesity? Well, 1, you're low in alpha-ketoglutarate. 2, you're low in vitamin B6. 3, you have a high NADH to NAD plus ratio. And 4, you're low in endogenously produced alpha lipoic acid. And I want to be clear, I've talked a lot about alpha lipoic acid as a supplement, and alpha lipoic acid will absolutely, especially the R version, R-ALA, will bring down this NADH to NAD plus ratio, and that's great. However, that ALA that you take as a supplement cannot be used as an enzymatic cofactor. For that, you have to produce endogenous ALA. And so there's 2 different reasons that we need ALA around. One is just to reduce this ratio. Another one is an enzyme cofactor. And so the limiting factor, I think, I'm sort of guessing, but I think that one of the limiting factors for producing this ALA is a lack of methyl groups. And that all, of course, goes back to the aryl hydrocarbon receptor, and you can watch videos about that. But I do think that to produce more of this ALA endogenously as an enzyme cofactor, you need methyl groups. That means choline, that means betaine, that means SAMe. And so there's not a ton of research on alpha-ketoglutarate in humans. There is some, but this is in mice. And you can see this is mice on a high-fat diet. And this blue line are mice on a high-fat diet supplemented with alpha-ketoglutarate. So you can see it didn't completely reduce the weight gain from the high-fat diet, but they are indeed leaner. And interestingly, you can see this is the metabolic rate. So this is from that same study. The red mice are mice on this high-fat diet, and they have the lowest metabolic rate of any group. And the blue mice, the ones given the alpha-ketoglutarate, there is a significantly higher metabolic rate there. And they again use the UV camera on the mice, which is pretty cool. And this is these are the different colors and how they correspond to temperatures. It's a little hard to see, but white is actually the hottest color. And so you can see here's the mice on the high fat diet. They're kind of red, but the ones on the alpha ketoglutarate, 1, you can see that they're leaner. And 2, they have a lot more of this white hot areas on their body. And so you can see the mice on the high fat diet have the lowest body temperature of any of the mice. And the alpha-ketoglutarate goes a long way to putting that closer to normal. And you can see the rectal temperature, the high-fat diet, very low rectal temperature. But if you add back the alpha-ketoglutarate, they have nearly the same temperature as the mice on the normal diet. This is one more study. This is in rats. And so what they showed here is that, so here's these amino acids, right? So here's valine. And this is this is just a low-fat control rat on a normal rat diet, right? And so the level of valine, that's one of the branched-chain amino acids, is 184. And then when you put them on the high-fat diet, it goes all the way up to 233. When you add back the alpha-ketoglutarate to that high-fat diet, it goes back down to 181, which is interesting because that is exactly the same as the control level, right? And so that's cool. And so these are the other 2 amino acids, or these are the other 2 branched chain amino acids, leucine and isoleucine. And you can see in both cases, the alpha-ketoglutarate restores the branched chain amino acids back to the same amount as the control group. And they're, of course, both higher in the high-fat diet. So that's interesting. And lastly, what this study shows is that the alpha-ketoglutarate restores insulin sensitivity. See, so these columns line up the same way. The low-fat control, their insulin sensitivity is 1,000. This works in an inverse way. So if your insulin sensitivity scores 1,400, that means you're insulin resistant. And the alpha-ketoglutarate puts their insulin sensitivity back to normal. And so wrapping this up, and so I'm now offering alpha-ketoglutarate. It's actually through Doublewood Supplements. They're a great manufacturer. They already had a product on the market that you can't find in a ton of places, but it works great, I think, with the disodium succinate that I already have on there. You know, we know that branched chain amino acids are elevated in torpor and obesity. This is probably a mechanism which the torpid animal uses to preserve lean body mass and also to cause it to become insulin resistant, which if you're a hibernating animal, that's what you want, right? But as a walking around human, maybe not so much what you want. And so these branched chain amino acids are catabolic, right? They will cause you to add lean mass tissue. And when you look at obese humans, they have a lot more fat. They also have more lean mass, right? And that's probably because these branched chain amino acids are just, bulking them up generally, right? So they're catabolic, and they likely are involved in causing insulin resistance. And one of the limiting factors to breaking down those branched chain amino acids is Alpha-Ketoglutarate. Like I say, you can get that now at fireinabottle.net/shop. And if you're interested in supplements, what would I take along with Alpha-Ketoglutarate? Well, sort of the same things I always talk about. Our alpha-lipoic acid is a great one. That helps get that NADH to NAD plus ratio down. It helps get things deacetylated. That's what you want. Sources of methyl groups: betaine, choline, SAM-E. I would take those, and perhaps activated vitamin B6 PLP. The active form of B6 is very good. That is it for now, guys. Go out there, burn some branched chain amino acids, and I'll see you next time.